Welcome everyone to this new Gen webinar sponsored by Hera and Demeetra entitled Cas-CLOVER, the clean alternative to CRISPR-Cas9. I'm Jeff Buguliskis, technical editor for Gen, and I'll be your host today for this webinar. Though many of us have been stuck indoors for several weeks now, cut off from our normal lives, most of us are still pretty aware of the potential that gene editing systems such as CRISPR-Cas9 possess for therapeutic intervention. It seems like almost every week, a new use for CRISPR appears in the literature, validating the true power of this technique. However, as with any new technology, improvements can be made to streamline the development process. In that vein, researchers have created the novel and truly dimeric Cas-CLOVER system, which differs from the traditional single-guided Cas9 and dual-guided Cas9 Nuclease methodologies. The Cas-CLOVER platform has some unique advantages, as it demonstrates high fidelity and efficiency with no discernible off-targeting. Let's meet our presenters for this webinar, who are going to tell us about the implementation and validation of Cas-CLOVER, as well as its use in various human and animal cell models. Dr. Setyen Jamling is the Vice President for Research and Development and Gene Editing at Hera Biolabs. Dr. Jamling will provide us with some background information on Cas-CLOVER and show some data of Cas-CLOVER efficiency on various cell types. Next, Dr. Maximilian Richter, a senior scientist for immuno-oncology at Posada Therapeutics, will tell us how the Cas-CLOVER system is used in the generation of healthy, donor-derived allogeneic CAR T cells, while overcoming consistency and variability challenges known to plague the scale-up of this therapeutic model. But before we begin, I want to remind the audience to stick around for the Q&A session that will begin right after the last talk. Don't be shy. Send in any question you might have for our panelists. It's really easy. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. We'll try to get to as many questions that we can. Okay, with all that said, I'm now going to pass the presentation over to Dr. Jamling.
Hello, everyone. Thanks for joining us today. My name is Setyen Jamling. I'm the VP for R&D and Gene Editing at Hera Biolab. It is my pleasure to introduce the Cas-CLOVER gene editing system shown in cartoon format here. Cas-CLOVER utilizes a catalytically inactive dead Cas9 fused to a CLOVE51 nuclease domain, and it works as a pair of monomers recruited by a pair of guide RNAs to introduce targeted mutations. Hera Biolabs is a technology company with products and services surrounding our proprietary gene editing technologies. The piggyback transposase system, the Cas-CLOVER gene editing system, about which we'll talk at length today, and the Tau-CLOVER system, similar to Cas-CLOVER but built around the TALEN architecture. We make these technologies available under a research license. We also use these technologies to offer custom genome engineering services to create cell lines and custom animal models. We use them also to create our own proprietary genetically engineered research models, one of which our platform model is the SRG rat. It is a RAG2 IL-2 receptor gamma double knockout immunodeficient rat, similar genetically and phenotypically to the NSG mouse, which many of you are familiar with. Using these technologies and platforms, we also offer contract research services. Again, as I mentioned, the oncorat being immunodeficient is highly amenable for human xenograft studies. So we do a lot of preclinical oncology studies, drug efficacy work, and model characterization, validation, and such. To understand Cas-CLOVER, I'd like to show you a cartoon depiction of how this system works. Shown at the bottom here is the presumptive genomic target locus. And as with any other gene editing tool, we start by introducing our reagents into the cell. Here we are transfecting the Cas-CLOVER mRNA along with a pair of guide RNAs to guide it to the cell, to the target locus. Once introduced into the cells, The mRNA gets expressed into Cas-CLOVER proteins, which then complex with the guide RNAs. This complex is then recruited to the target locus, just as it happens for CRISPR-Cas9. And when both subunits are properly targeted to the locus, it leads to dimerization and activation of the CLO51 nuclease domain, leading to targeted gene disruptions. The idea behind Cas-CLOVER was to combine the efficiency of the CRISPR-Cas9 system with the specificity of the TALENs. CRISPR-Cas9 is an easy-to-use, highly efficient gene editing system. You only need to design one guide RNA to get efficient cutting at a locus, but there are some concerns about off-target mutations caused by this system. TALENs, on the other hand, are highly specific, requiring two monomers to bind to introduce the cut. But it is hard to build, and it does not have the efficiency of the CRISPR-Cas9 system. Since Cas-CLOVER is recruited to the targeted locus just as Cas9 is by guide RNAs, our hypothesis is it should have the efficiency associated with the CRISPR-Cas9 system. But since it requires two guide RNAs to work in pairs to recruit two independent monomers and then subsequent dimerization of the nuclease domain before it can introduce a cut, it should have the same specificity as the TALEN system. And today I'll share some data with you to support both these hypotheses. We started by testing various fusions and guide RNA orientations using a dual reporter assay. The dual reporter plasmid has a constitutively active RFP fused to a GFP that is out of frame and off. Between these two reporters, we clone in our targeted locus. So in its native format, the RFP is on and the GFP is off. If Cas-CLOVER correctly targets and introduces a cut in the targeting locus, we would expect microhomology repair to put the GFP in frame with the RFP, thereby turning it on. So without cutting, RFP is on, GFP is off. With targeted cutting, RFP and GFP is on. So here, I'm showing what we settled on after various different testings. And the way Cas-CLOVER works best is if the two guide RNAs are oriented in the PAM's out format. Here's the left guide RNA PAM, and here's the right guide RNA PAM side, flanking the two guide RNAs that way. And the distance between these two guide RNAs, the gap, it works, we tested between 14 to 40 nucleotide gap, and shown below is the cutting efficiency we saw by this assay on a plasmid. So Cas-CLOVER works in this format over a wide range of gap distance, anywhere between 15, 16 nucleotides to about 30, 31 nucleotides there. You get efficient cutting. We settled ultimately on a gap distance of 16 to 20 nucleotides for two reasons. One, as you can see, that's the region we see the best efficiency of cutting. And two, because using this gap distance allows us to take advantage of all the guide RNA design platforms already available and that you are already using. This gap distance works well with all the paired Nuclease guide RNA design platforms. So any pair that you design using those platforms fits in seamlessly with the Cas-CLOVER technology. Following the plasmid reporter assay, the next step was to test Cas-CLOVER for targeting genomic DNA. Working with our sister company Poseida Therapeutics, Cas-CLOVER was used to target surface marker genes in human T cells, and the results assessed by flow cytometry. Starting from the left, mock transfected control cells all display T cell receptor alpha fine. Both the left and the right guide RNAs are efficient at recruiting Cas9 to the locus, leading to TCR alpha disruption. When you introduce the left and the right by themselves, single guide RNAs with Cas-CLOVER, we get no disruption of the receptor expression comparable to negative control. But when you introduce both guide RNAs as a pair with Cas-CLOVER, now you get efficient disruption of the TCR alpha expression that's comparable to the results we see here for CRISPR-Cas9. Same data is shown in graphical format here for TCR alpha. We also checked various other surface receptor genes and shown here, such as beta-2M, and the results were similar across the board, where CasCLOVER requires both the guide RNAs to be efficient at disrupting expression. So this supports our first hypothesis that CasCLOVER efficiency is comparable to CRISPR-Cas9 system. Next then was to test our second hypothesis that CasCLOVER should have little to no off-target activity. Shown along the bottom here is all the different ways we could think of that CasCLOVER might produce an off-target effect. Either the left guide RNA by itself binds an off-target site, thereby recruiting the whole complex to introduce a cut site there. Same case for the right guide RNA, where the right guide RNA binds an off-target site, thereby recruiting the whole complex. Obviously, if there was a left guide RNA off-target in close proximity to the right guide RNA off-target, you could think of getting the complex recruited there to introduce an off-target cut. Then the other way we thought was if there were two off-targets for the left guide RNA in close proximity, or the same for the right, you could think of it might introduce a cut there. Based on these possibilities, we worked with KBiobox, a bioinformatics company, to predict all the possible off-target sites for CasCLOVER in the genome. So shown along the bottom here are all the predicted off-targets for in the genome. A group of off-targets for the left guide RNA. Here, two left guide RNAs, left and right guide RNA combinations, just a single right guide RNA, and two right guide RNAs. Shown in the middle here is the actual on-target site. Along here, in blue lines, is the background mutation rate we saw in the mock negative controls. So the dotted blue lines here, two dotted blue lines, are the background levels plus or minus 1 standard deviation. In solid blue is the background level plus or minus 2 standard deviations. Shown in the reddish brown bar graph here, corresponding to the values on the y-axis on the left, is the mutation rate detected at each of these sites. Again, on the actual on target, we get highly efficient cutting, close to 70% for TCR alpha locus, whereas for all of these different combinations of predicted off targets, there is virtually no different from a negative control. So we couldn't detect any off target activity for any of the predicted sites above background levels. The last bit of information on this graph are these blue dots up on top, which represents the depth of coverage or the number of reads generated for each of these locus, corresponding to the numbers of reads here on the y-axis on the right. So for each locus, we generated anywhere between 50 to over 100,000 reads. And at that depth of coverage, again, there is no off-target activity detected at all. So this supports our second hypothesis that CasCLOVER, because it requires two guide RNAs to recruit correctly and dimerization of the CLO51 nucleus before it can introduce a double-stranded cut, there is no off-target activity detectable with this system. And we have looked at other locuses, right, surface markers with it, and they virtually across all targets tested, there's no off-target activity. We have also validated CasCLOVER in various other mammalian cell lines. Shown here is the editing we did in CHO cells, which is an important medium for bioproduction of human therapeutics. We were targeting the GS locus, which has been shown to be important for efficient expression of therapeutics. So And I'm just showing an early condition versus a later condition, playing around with different ways to introduce CasCLOVER to increase their efficiencies. So as you can see at the lower end, we were getting about 15, 20% mutation rate, which we can bump it up to well past 40%. Another one I wanted to highlight here is within a condition, we can do serial transfections to increase our efficiency. So in the previous slide, we showed that we really have no concerns of off-target activity. So then for difficult to target locuses or cell lines, we can be confident about transfecting them serially without any off-target concerns. So this is the first transfection for this group of cells where we got close to about 30% mutation rate. We let the cells recover 3 to 5 days and hit them again with the same reagents. And now in this pool, we have a mutation rate well above 40%. So what do the mutations actually look like with CasCLOVER? Here, the top line here is the wild type reference. Again, the 2 guide RNAs in the PAMS out format. Left guide RNA binding site, right guide RNA binding site. And when recruited properly, the cut gets introduced in this space region in the middle, and that's where you see the mutations and deletions happening with CasCLOVER. Not only in mammalian cell systems, working with Demeetra AgBio, we have verified CasCLOVER activity in plants also. So here they were targeting the RDR6 locus in tobacco plants, which has previously been shown to be important for transgene expression, with the knockout getting higher level of transgene expression. And shown on the bottom here, again as before, the binding sites for the left and the right guide RNA and the different type of mutations that were generated using CasCLOVER. Another system this has been tested in is in the yeast, another important medium for bioproduction. Here we were targeting the ADE2 locus, which leads to red pigmentation of yeast on these plates shown here. And if you on the left here, as you can see, we can get knockout efficiencies to about 80%. So very efficient knockout in the yeast system also. So with that, I will end here. Hopefully the data I've shown today helps convince you that CasCLOVER is an efficient gene editing tool with efficiencies comparable to CRISPR-Cas9 and with specificity comparable to the TALEN system. Thank you.
Thanks, Aten. That was a great way to start off our webinar and some really nice insight into the CasCLOVER system.